our esteemed speaker, Dr. Karthik Rajendran. Over to you, sir.
Hi, thank you very much. Thank you all for the participants for being here. I would like to thank my formally thank Chennai Research Foundation for organizing this particular 1 week workshop on algal biorefinery and biotechnology. This is the first of its kind. It is something new, and I am excited to listen the other talks as well. Today, I'll be one of the speakers who will be talking about what we do at our lab, the Net Zero lab at SRMAP. I wish Chennai Research Foundation could come in and bring more useful, more interactive programs like this. Maybe they can conduct workshop, hands-on training, and some other things related to the cutting edge area of what is happening globally related to sustainable aspects. Having said that, let me start over my presentation. So it will approximately take about 1 hour or so. So after that, I can take questions, or if you need anything clarification in between as well, I'll be happy to go with it. Just let me know if you can see my screen.
Yes, sir.
Is my screen visible in full mode, full screen? Is my screen visible in full screen? Hello?
Yes, sir. Yes, sir.
Okay. So today I'm going to talk about microalgae and machine learning and how we can use it to treat wastewater. So what we have been doing over the last 5.5 years here is I think, is it fine now? Can you see the full screen?
Yes, sir.
Okay. So over the last 5.5 years, we have been working with respect to how we can treat our wastewaters more effectively, but not only from a conventional point of view, but also from other aspects of looking at the wastewater. So the topic I have chosen today to present is microalgae and machine learning and how to treat it for wastewater. Currently, I'm working as an associate professor at SRM University, Andhra Pradesh. We run a lab called the Net Zero Lab. We work on decarbonization technologies, net zero emissions, biorefinery approaches in a not a traditional experimental way, but we look at it from an unorthodox, non-linear pathways to study the theoretical aspect, and we try to solve the problem at a systems level. So if you take the wastewater in India per se, right? So there are about 72,360 600 million tons of wastewater sewage is produced every day. Only 1/3 of it is treated, and almost only 2/3 of it is collected. What happens is almost the remaining goes to the river streams and other places untreated. That's where we start to see the pollution which is happening. Besides that, there is another problem which comes is India accounts for the second largest fertilizer consumption. Our food requirements is quite high, and we spend more than 15,000 crore per year in terms of importing the fertilizer. And every kilogram of fertilizer which we consume takes about 23 kilograms of carbon dioxide equivalent. Fertilizer industry is one of the heavy carbon industry which takes the large amount of carbon dioxide because of the way in which we produce ammonia, etc. So there was a question, why can't we look at the sewage as a source to produce fertilizer? That is where we started this particular journey over the 5 year back. And what image which we are seeing at the bottom is actually the integrated biorefinery part which we are trying to develop. This is a just a 3D image for that. So when the wastewater comes, if we pre-treat, the algae gets cultivated. From the algae, we get the after that we have the clear water, and then the algae is converted to value-added products and fuels, so that we produce a sustainable circular economy as a supply chain. So if you look at wastewater, this particular graph on the x-axis, you are seeing the state-wise GDP. So here, all these points corresponds to the GDP of different states. And on the y-axis, you look at the wastewater generation in million liters per day. So you can see a strong correlation that the state which has high GDP is also producing high urban waste water, which means that as the economic prosperity improves in a particular state, the consumption is also increasing, right? So not only that, the population versus wastewater also increases, so there is a direct correlation. So you can see the correlation here as well. So what does this tells is the more we are and the more economically prosper we are, the more wastewater we are going to produce, which means the more problem we will have to solve as well. So that is something which we have observed. So with that particular thing in mind, first we wanted to check whether the urban sewage, can it be a useful resource for us? Is it something, you know, um, necessary, or can we do, is it actually a resource or not? That was the question which we had in mind. With that, we started something known as the resource estimation. So what we did was we tried to take the urban sewage. As I mentioned earlier, only one third was getting treated and two third was not getting treated. We tried to do a theoretical calculation on the nutrient recovery with using different approaches. I'll go into the details soon. So when you have the nutrient recovery approaches, you can see that the biofertilizer which comes out of is 0.38 million tons per annum. Of course, it can also reduce the emission because it is from the wastewater resource. So from there, you are trying to capture the uh, uh, fertilizer. So which means you are avoiding the generation of fertilizer from the synthetic resource. So you are avoiding 53% of emissions. And the imports, which is like, as I mentioned in my first slide, the amount of we spend on importing fertilizers, we can save up to 110 million US dollars per annum. This is only with respect to sewage as a resource, not the entire wastewater which is produced in the country. So after we did that one, we wanted to check which method is actually, from an energy point of view, which method is actually beneficial. So here you see on the right hand side of this particular thing, on the x-axis is the wastewater treatment methods, and on the y-axis you have energy consumption in kilowatt hour per 1000 cubic meter of wastewater. So all these are the conventional technologies. You have your SBR, UASB, FBR, all those activated sludge process, etc. And then what we did was we tried to compare it with the other kind of methodologies. So this particular A is actually STP plus chemical precipitation and ion exchange. Then C is only fuel cells and D is STP plus microalgae. So you might wonder why microalgae is actually having on the very high side of it and what is in the point. That's absolutely right. What we did was we added first STP, then microalgae as a tertiary treatment options. So because of which the energy consumption was quite high. But one thing which we observed noticeably here was the way in which fuel cells look at it. Fuel cells we did not treat along with STP. We take it as a sole treatment method, and we observe that if you are recovering nutrient and treat wastewater at the same time, then you can reduce emissions quite a lot. So that is where you could see one of the lowest in the trend. Then we thought, why don't we use this particular technique in microalgae instead of combining with STP? So But there was one problem as well with respect to the fuel cells, though it was quite less in terms of uh, the emissions. But what happens is fuel cells is not a scalable technique. It has its capacity wise. It is up to now the maximum reactor size it can go is up to 50 cubic meter, which means the scalability is an issue. So what we tried was we tried to look at why don't we use microalgae in this particular system. Then we see whether we can recover nutrients or not. So this is a continuation of the previous slide where we have the existing wastewater in different techniques, chemical precipitation, ion exchange. So you can see what is the cost and energy it takes up in terms of this. But when you look at it from a microalgae point of view, you get about $70-80 per cubic meter. And the energy consumption, as I mentioned, it is quite high because of the combined treatment systems along with the STP. So that's why you have from the microbial fuel cell, you have a direct arrow, and for the other treatment systems, it is coming after STP. So this is the way it looks at. Okay, now we wanted to look from a theoretical point of view. We understood that the resource is valid, and from the energy consumption we look at, or from an economical point, we see that microalgae or microbial fuel cells could be a good option. Then we wanted to study the entire system as such. So that's why we started with working on a life cycle assessment. Life cycle assessment is a technique where you look at what are all the impacts that has on the entire life cycle. This can be done in different ways. First step will be actually to do the goal and scope definition, then the inventory collection, then you have the life cycle impact assessment, and then the outcomes, which comes as what we get as the different indicators, which is usually the midpoint indicators. There are 17 to 18 in number. We use the recipe 2.1 method and echo invent database for the same, and we use SEMA Pro software. So if you look at this particular thing, so we define to reduce to produce 1 ton of 1 kilogram of nutrient, what is the thing, or to treat 1 cubic meter of wastewater, what is the energy and the emissions it occur. So that is the functional unit we take into the picture. This is the overall system boundary. So what happens here is a wastewater which comes into the system, then first it goes to the screening and grid chamber, and then you have the primary treatment, secondary treatment, clarifier. Then from there you have the thickening, thickening effluent, dewatering. Then you have anaerobic digestion, decanter. So this is the typical STP process. And then you have the nutrient recovery systems. So we had, as I said, the 4 nutrient recovery systems. One is the chemical precipitation, ion exchange, microalgae. So from there you get the product, biofertilizer, crude, etc. And whereas after this nutrient recovery system, the effluent is discharged. And here as well, you get the dry sludge as well. In case of only fuel cells technique, the wastewater goes in and then it goes into the fuel cells directly. And from there, it goes to the fertilizer or the product. So this is the scenarios which we have done. So first scenario was only the base case scenario or the business as usual, where the conventional wastewater treatments is the STP process. And then you have scenario 2 with fuel cells, scenario 3 with STP plus chemical precipitation, STP plus ion exchange, and STP plus microalgae. So if you look at the overall global warming potential is the one how much grams of CO2 is being emitted to treat 1 cubic meter of wastewater. So if you look at this particular thing, a typical wastewater system, the sewage treatment plants releases about 400 grams of CO2, while microbial fuel cell was an overall negative emission technology about minus 234. So this particular plus on the 304 is the one which is produced from the process, and the minus 538 is what it get absorbed from the process because of the emissions reduction overall. So likewise as well, because this minus 96 and minus 155 in the chemical precipitation and ion exchange is because of the nutrient which we have recovered. So that is something which reduces the overall impact when you compare with the conventional wastewater treatment. Whereas for microalgae, as I mentioned earlier, the negative emission is there, but since because we are doing it after the treatment, the overall energy consumption is shoots up, which I have showed you in the earlier slide. That results in the overall very high global warming potential. The same scenario is also applicable in terms of terrestrial ecotoxicity. Terrestrial ecotoxicity is something which looks at the amount of toxic elements that is being discharged to the your different streams, air, water, and land. So that is something which you consider here, and you can see that overall the microbial fuel cells seems to be better. But as I mentioned earlier, the issue with microalgae is the scalability. You want to see a technique which is scalable, which can be uh, multiplied. So for that particular thing, we wanted to use the microalgae. In microalgae, the problem was most of the energy was consumed in aeration and decanter. So if you look at the overall global warming potential when it comes to the comparison, when it comes to commercial fertilizers which are available, you can see this A, B, C, D, urea, DAP, MAP, NPK, Struvite. Those are in these particular areas, whereas E, F, G, H, it is all the fertilizer which has been produced from the different treatment methods. A difference of reduction from 56 to 98% was achievable. If you have the global warming potential, it was achievable up to 56 to 98 percentage. So that is something which gives a significant reduction from how we are importing fertilizer versus how we can actually take the look at the fertilizer in a different way. So this was the petrochemical fertilizer, and this is the biofertilizers. Okay, now we did the assessment. We know that in life cycle you can reduce the potential. However, you are not sure of go ahead with the microalgae, or you want to do something else. So what we did was we take three different wastewater. Urban wastewater, we compared the university wastewater, municipal wastewater, and dairy wastewater. The characteristics of these two are almost similar. However, in the university, from the university, we take the wastewater which was having a little bit of homogeneity. Whereas in the municipal wastewater, it is much more heterogeneous because it has various sources where it all get mixed up. The dairy wastewater is an homogeneous wastewater. However, it has a very high protein content. So what we did was we compared these two wastewaters in these two species for growing up the algae. So there was air which was pumped in for the growth of algae for aeration. And from there you can see the biomass growth which we obtained was 725 to 770 milligram per liter. The COD reduction of the wastewater. Remember that this is the not the wastewater which is after STP, it is the raw wastewater which we have taken. The COD reduction was 74 to 94 percentage was obtained, whereas the nutrient recovery was seen up to 85 to 97 percentage. So this is the experimental setup. You can see about what we have been doing. So you have the wastewater. So we have the control as well. And then after adding inoculum and after 9 days, so you can see there is a color which is getting changed. It's getting darker and darker. So the more, darker it is, the higher the cell growth as well. Then if you look at the biomass recovery, what happens is if you look at the dry weight cell on the y-axis and the day of growth on the x-axis, and this is the control university wastewater, municipal, and dairy wastewater. The dairy wastewater mostly out surprises the other part because of the high protein content which it has, whereas followed by municipal and university wastewater. The same is the case of in both Senidesmus and chlorella. However, Senidesmus was much more having a higher production rate when it compared to chlorella species. So what we found in this particular study was there is inaccuracies in the estimation of cells and then it was a very less cell concentration. And this technique is also like to maintain, it is very capital and laborious. So we wanted to find a biomass quantification method through different approaches. Of course, there are different approaches available. You may all be aware of dry weight system, turbidity, cell counting, optical density, or cytometric systems. So out of these systems, we wanted to look at a mechanism where it is easy to measure the cells so that we know whether we can go ahead with this particular technology or not. So we wanted to integrate the part of the laboratory of AI work to predict using AI to algae. So we looked at different kinds of mechanism, and we wrote a paper on AI and algae. Can we work together? So we looked at different machine learning algorithms, how they work, where we should use, what is the use of the machine learning, etc. So this raises the question for the interest of audience, if you are from diverse background, just a question: what is machine learning? Machine learning is an area of AI where we use different algorithms to learn the pattern and measure what is going to happen or what we could have been achieved. There are three ways of doing it. One is the supervised learning, unsupervised learning, and reinforcement learning. Supervised learning is like how a teacher teaches the student. So someone should be there to tell this is right, this is wrong. Whereas unsupervised learning is the student studying by themselves. They learn from the book, read, and etc. Reinforcement learning is you don't have a formal education, but you are learning through trial and error. You try something, it works. If it doesn't work, you learn from the mistake and then you redo it again. So this brings the question when to use machine learning. Usually, typically, for all the experimental design, we used to have the statistical methods, and we use different kinds of tests, different mechanisms, which has been highlighted here: t-test, ANOVA, chi-square, regression, etc. But on the other hand, from the machine learning side, if you want to do a prediction, then you can go for labeling the data and classify. That becomes the supervised learning. In prediction, you want to do quantification, you can go for regression. If you are looking for more insights and data analysis, then you can look at unsupervised and reinforced mechanisms. So you can do a lot of things with respect to adding, changing the data, changing the images, etc. So based on this technique, we have initially used from the statistical point of view, and then we have shifted our focus towards the machine learning point of view. And I'll tell you why. The reason is when it comes to the statistical thing, you can predict, but then you cannot forecast. So the difference between prediction and forecast is prediction is this is where you want to go. But forecasting is you don't know where you have to go, but that should be the end point. So that is something which statistical methods, sometimes it is difficult to achieve. And they have its own limitations with respect to the data sets which you are going to handle. So with respect to machine learning, you have different steps. The first step will be the data collection, data cleaning, feature engineering, machine learning model selection, and training and evaluation. So once you have the data, in this case, the data can be from any source. For example, with respect to the from the experiments, it can be, and from the observations, it can be. The way in which we design the experiment is also matters with respect to the way we collect the data. There is a completely balanced approach to the experimental design. So if you are going to do that, how the data collection is going to be, what are all the parameters which is relevant. Okay, now I have the data collection. Does it mean all the data needs to be used, or do I need to do a data cleaning? It's not, maybe some observations, there would be a human error, there would be a system error, there would be an equipment error, or the way in which I have the data itself has to be transformed. So in those cases, I need to clean the data and prepare the data for analysis. After that, you get the feature extraction. In feature extraction, what happens is you look at what are all the factors which are critical for evaluation. Which factors contribute the highest to the prediction of your particular variable. And based on that, once you do the normalization of the values, then you can see based on the errors and the R square, you can find which model is good, why I should select that particular model. And of course, the model has to be trained and tested, and only then it can be validated. So the need for prediction is uh, what happens in wastewater treatment with algae is, if you see the particular graph, this is the microalgae yield which we can get from a typical wastewater treatment systems. But if we can optimize the conditions, the input and etc., we can get up to 31% higher yield. Like from 770 milligram per liter, which we have reported, we have gone up to 948 milligram per liter by providing an optimistic conditions. Just we have to play with the conditions which we are providing to the growth of algae. So if you look at the literature analysis, artificial neural networks has the highest R square value, which has the good fit with less error. This particular chart talks about that particular thing where you have on the x-axis the error. So you have low error, high error, and then here you have low R square value and high R square value. So if you look at this particular quadrant, the top right quadrant, you can see that ANN has the more error and high R square value. So overall, the error wise, ANN was good with, and also the accuracy is also better. So based on this particular thing, we carried out a full experiment to run the wastewater system using these different parameters. You can see phosphate, daylight, temperature, nitrogen, nitrate conditions, what is the input COD, pH, carbon dioxide, etc. Then the output parameters were chlorophyll A, B, carbohydrate, lipids, protein, pH, microalgae yield, etc. And then we did a prediction model based on that. And not only prediction, but also an optimization model. So here you can see the model performance. This particular leading line is called as the perfect fit line, which means the value is 100% accurate. And these are the training data set. You can see in terms of training, we have achieved up to 99% accuracy, whereas in the validation point, we got up to 97% accuracy. So how we train the data is like, if you have 100% of data or 100 values, you can use 80-20, 70-30, 60-40 like that. So sometimes the error which you get from the validation, maybe it may be over 50 or it may not be validated properly. So that is where we need to do a sensitivity analysis on this particular thing, whether we are getting the right fit or are we overfitting or underfitting the data. So based on these ones, you can see most of the model can predict the growth of algae with a quite a high accuracy actually. So based on this, we devise the optimized conditions. What should be the optimized conditions? So if you can provide this particular condition, as I mentioned, we can go up to 950. If you remember from the experimental setup which we have done, we have went only up to 770 milligram per liter. Now the question may come whether like can we give these exact inputs as such? Maybe not. But if we are trying to add some nutrient supplement in any of these component per se, and if we could maintain the parameter, then this particular result is possible. So this brings to the question of prediction is fine. We can predict what we have achieved. So prediction is something I am tracing this particular track back again. Whereas forecasting is something I am here but I have to predict what is going to come next. So based on this particular data trend, I am going to find out what is going to come in front of me. So this is something which we have started to work on it with respect to different algorithms which we are using, ARIMA, LSTM, XGBoost, and Prophet models. So here, if you look at this particular chart, so this is the experimental data, and here you have the different algorithms. So if you look at the overall, here you can see the inside thing, what is the deviation. Most of the model, if it has a more than 5% error, then the model was rejected. If it is less than 5% error, then that was considered for residual analysis, where we looked at the how the errors are being spotted, and from there we identify what is the best model. So if you look at the pre-processed data that goes into the model fitting to find out this particular curve. If you look at this particular forecasting, I was talking about the prediction where we had the accuracy more than 90%, but with the data set available and the non-linearity of the data, you can see that the R-square value has come down from 98% to 75%. So that is what has happened. But however, without the data point, still you could know the way in which the trend is going to happen. So the dotted line here represents the future prediction. The graph, it shows the biomass, while the light green line shows the training data. So you can see here, this particular point, the training data is stopped. There was no data after this point. So the dotted line predicted the same kind of movement. Of course, there was a little bit of deviation with respect to the accuracy point. And if you look at these residuals, you can see that over the course of time, in terms of testing, the residuals has also gone down. Here you can see a lot of variance that is happening, whereas in this particular area, the variance has come down, and you can see the distribution of residuals is a training and testing both follows a typical normal distribution curve. Okay, so now we have looked at how to predict algae, how to forecast algae. But then we have wanted to go deeper into the question. Can we look at what is the species, what is the genus, what is the type of algae, what is the morphology of algae? Can we predict that particular thing using our machine learning? So we did another experiment looking at the data collection for just by taking an image from a petri dish. Can you just or take a photo from your mobile phone and say, I want to just look at this one. I want to say how is the health of my reactor, just by doing an image analysis technique. So, and then of course we can also predict the biomass as well. So just by taking the picture. Earlier we were looking at the complete chart growth, input parameters, output parameters, etc. Whereas in this particular thing, we wanted to go little bit deeper to understand. So by default, if you see at this particular picture, you can see like this is a at the start of the culture, and you can see the darker it is, the more algae it is, right? So the gradual variation is observed through the color density. It was opened avenues for predicting using advanced techniques. So what we did was we took the microalgae in a petri plate in day 1, day 12, different stages. We took almost near to 1,000 images using the, we also measured in parallel the OD value in a spectrophotometer. And then one thing also which we did was the standardizing the image to have the same objective. We don't want to have a different pixel ranges, so we had the pixel as a height to width of 100 each. So when you look at this particular graph on the over right hand side, you can see that the color density increases over the course of time and how darker it is. So this is from the experimental value, and we wanted to measure the same. So there was a correlation that the number of days it takes is directly proportional to the concentration of the algae it becomes. So as the days increases, the algal concentration increases, leading to a darker green color. So here you can see the how the microalgae is looks at from a machine learning focus. So here you can see the leading line, and then you can look at the accuracy as at 98 percentage. The training data set and the testing data set was almost quite in favor. And you can see how the other algorithms works as well. It was put in dull in color to have a focus over here, but you can see the other things with respect to the R square values and error. So this particular model had the highest R square error with the lowest error. So the thing with respect to the focal point is how we take the image actually matters in terms of how we analyze the image. So if I'm going to take the picture in a different way, how I focus, out of focus or in blur focus, that is going to help me actually to get the type of image. So we have tried to tweak these kind of things as variations to our system to measure whether still the microalgae can predict or forecast the biomass. So if you look at this particular slide, we have considered different techniques. For example, like blurring. Blurring is something like you just smudge it. Contrast is changing the color density, or offline transactions, noising and denoising techniques. So if you look at the different methods which we have used and the R-square value, so you can see, for example, flipping the image has higher error, Gaussian blur almost or real sample. So there was not much considerable difference if you are using different techniques. In fact, the error values, when it increases, your accuracy as well goes down. So based on these techniques, we can see whether our measurement methodology is reliable and it is scalable. So we know now we can use the image analysis, but the question remains is, can we go even further to predict just by taking a picture? If I can isolate, what will be my species? There are different kinds of microalgae are there, unicellular, multicellular, and also the shapes, morphology, with the flagellae, structure, etc., etc. So can we predict this? There are certain challenges to do with respect to how, because the as I mentioned in the initial slides with respect to the classifying the labeled data is one of the critical challenge to train the model. And for an unsupervised or reinforced learning, it is of a little bit more tedious task to start with. So we are looking at different microalgae in morphological diversity with respect to the shape in terms of spherical or spiral, and then we are having microns, like looking at the size of the image, microns to millimeters, and as I said, whether it is arranged in unicellular or colonial type. So we are looking at these kind of different species to identify. Just by taking the image, we extract the different features of it, looking at it, the shape, size, and color. If it can enhance the accuracy and reduce the error, and which enables a real-time monitoring. So this brings to my almost close of my talk. If you look at the emissions per se, wastewater as a technology can reduce 53% of emissions if we can integrate proper recovery of fertilizers from it. Not only from an emissions perspective, but also from the perspective of economic savings, we can save a lot. This is 110 million is only from a sewage point of view. Of course, this is subject to the way in which we are collecting the wastewater, processing the wastewater. Then you can see the savings from an algae, how much we can earn, and as well as the nutrient recovery rate is quite impressive, from a 90 to 95% yield could be achieved. Not only that, using machine learning, we can increase the yield in which we perform experiment. we can optimize the conditions, and we can get even yield. So of course these numbers may change based on the yield which you are going to get. And then if you look at the accuracy of the machine learning, it is almost like 95 percentage. So just to give you the summary of uh, what I have presented over in the last 30 or 35 slides or so, I'm just summarizing it in this particular one slide. If you look at 2019, that's where we started to work on the resource estimation, estimating the carbon dioxide, what is the important import avoidance potential. Then during COVID, we started to work on, of course, we have been hampered with respect to the usage of labs. So that's where we started with the mass and energy balance. And then we looked at designing different processes, doing the theoretical calculations to understand how the mass and energy balance works on different wastewater treatment systems. What is the impact of that one in the energy and economic assessment? Then we carried those. The critical thing when you want to do the life cycle assessment is the way in which you are going to get the mass and balance data. So that's where we have actually calculated each of those mass balance data in our paper, and we took those things to the life cycle assessment. So once you have the life cycle assessment done, then you look at the really different picture of what we have been doing. Does it make sense to the real world industries and from a sustainability point? The question of sustainability is how sustainable actually the sustainability is. So we wanted to answer those things, and then we wanted to prove it with the experimental validation. Once we started the experimental validation and when the algae was growing, then we got into different kind of challenges with respect to after the post analysis, the laborious work it takes. So we wanted to develop methods and techniques wherein which we can reduce the human effort, and per se we can look at uh, the holistic point of prediction of microalgae or the treatment of the systems. So that's where we started to work on AI, and we have done a lot of work on this particular area using different models. Then we started to once we predict, we had the problem of okay, like this is working, but can we forecast it? So that's when we started developing on time series. The thing with respect to time series is at a static condition, it is easy to predict a point. But then with respect to the varying of time, like for example, at 8 o'clock, okay, I am hungry to certain level. At 8:30, my hungriness may be different. But can you forecast that particular thing? That is something which is actually a difficult thing to sort it out, because there are several dynamics which involves in respect to uh, the way in which we look at time as a component in terms of measuring the data, because at every data point with respect to time, the data changes. So that is something which we have done, and then we try to quantify the image using the machine learning model and analysis and OD measurement. So in our group, we work on various different aspects. I'm highlighting now. Karthik is one which is working on. There is another Karthik who is working on Karthik, who is working on the wastewater treatment, and Rajalakshmi is one. She is the one who is working on identifying the species using machine learning. And of course, we have other verticals where Girisha, she is working on looking at the environmental sustainability governance structure, policy perspectives, how to bring in SDGs, the theoretical framework behind it. One interesting question she is trying to address is what factors motivate students to move from one state to another to study. And then we have Rahul Sudhakaran, who are working on looking at the micronutrients and its effect in the anaerobic digestion, and can we bring up a commercial product just if you can have a one tin can, you add it to the system so that you do not need to worry about any micronutrient deficient in your process. Deblina, she is working on the AI models for mapping the crops productivity, etc. And Varshini, she works on cassava biorefinery. She look at the strategies to uh, make the cassava biorefinery depending on the market rate, how we have to choose the pathway. So that is something she is looking at, like market optimization kind. And we have Rahul and Vignesh, they are working on decarbonization of pathways. So we are looking at cement industry and as well as steel industry. We have already published a paper in decarbonization of sponge iron industries, and Rahul currently have started with the cement industry per se. So I thank the Chennai Research Foundation and the audience who have been patiently listening to me for the last 1 year, 1 hour or so. And if there are any questions, I'll be happy to take it up.